topic for today is translation. You know what is translation? We have already discussed. It is the polymerization of amino acids to form protein. Okay, so our characters are expressed as forming proteins. A DNA can express our character by forming a particular protein. You know that the gene expression is by forming a protein. For that we have discussed one gene, one protein hypothesis just mentioned, and one gene, one polypeptide. And recently we have discussed the cistron concept also. Okay, so translation process is the process of polymerization of amino acids to form a protein. And you know, it is based on an mRNA template. Okay, the genetic information in the mRNA as triplets is copied to a chain of amino acids. That is what is translation. Okay, come to the topic translation. See, as a preliminary step of translation, there is, you know what are the requirements of translation? You know, for translation, what is translation is mRNA is copied to a chain of amino acids. Okay, the transfer of genetic information from RNA to protein is known as translation. For that, the first requirement is an mRNA. Okay, that we have, the last class we have discussed how it is copied. Okay, by transcription. And next requirement, where this process takes place, where the translation takes place in our body. Okay, within a cell, okay, inside the nucleus, the DNA transcribes to number of RNAs. Okay, all the types of RNAs, tRNAs, mRNAs and rRNAs are produced here. And this RNAs, okay, from the nucleus, suppose this is an mRNA. From the nucleus, this mRNA is transported to what? The cytoplasm and mainly to the protein synthesis machinery. The translation machinery is ribosome. So understand, from the nucleus where the RNA is synthesized, from that place the RNA you have studied, it is well processed, capping, tailing, everything. Then it is transported to cytoplasm. Okay, now the RNA is in cytoplasm. Okay, now the RNA is in cytoplasm. The cytoplasm contains what? Ribosome also. You know, one of the cell organelles, ribosomes. And earlier, at first it will be remaining as small and large subunits separate. Okay? Normally, within the cytoplasm, the ribosomes remain as its smaller subunit and larger subunit separate. Then we will discuss what happens at the time of translation. Anyway, understand, the translation machinery is what? Ribosome. So the first requirement is an mRNA. Then what else is required is the site of translation, where the translation takes place. That is what is a ribosome. Then what else is required for translation? You have already studied a transfer RNA is required as an adapter. To bring every amino acid, there is a requirement of a tRNA. Okay, so a tRNA is required. Okay, in the ribosome, rRNAs are also required as catalyst. For translation to occur, tRNA is required, rRNA is required, mRNA is required, and where the process takes place is inside the ribosome. And one more thing, here for the translation to occur, number of amino acids are required. The amino acids should be joined. Okay, the amino acids should be joined to produce protein. So, once again, come back. The essential requirements for translation. The site of translation, where it takes place is ribosome. So, the ribosome is must. Okay, and the process also requires mRNA, the template which should be copied, then transfer RNA which brings the amino acid, then ribosomal RNA which acts as the catalyst which is present in the ribosome. The ribosome consists of rRNA that is required as a catalyst. We will discuss it. Then also we require different types of amino acids. Okay, these are the essential requirements for translation. So tell me, which is the site of translation? Site of translation? It is ribosome. mRNA, what's the role of mRNA? Template. Okay, should be copied. Then tRNA, what's the role of tRNA? Bringing amino acids, to bring amino acids. Okay, specific amino acids. Then next, ribosomal RNA, rRNA in translation. Where is rRNA present? It is present inside the ribosome. Okay, these rRNAs are having catalytic role. They act as catalyst. Okay, 23S type rRNA. Then when rRNA act as catalyst, it is not as, it is what a catalyst means. What a biocatalyst is, an enzyme. Okay, when rRNA act as enzyme, it is known as ribozyme. Okay, so keep this word in mind. Ribozyme. What is a ribozyme? You know, most of the enzymes are proteins. Enzymes are made up of protein. But here we have discussed enzymes are also made up of RNA. When an RNA functions as an enzyme, it is known as ribozyme. Okay, so what about ribozyme chemical nature? Chemically, ribozymes are not protein but RNA. Okay, then come back to the process of translation. We have discussed what are the requirements, and you know amino acids are present. Okay, tRNA is there. Then just understand, translation means what? Process of production of protein. Okay, that means many amino acids are joined. You know this amino acid, one amino acid is joined to next amino acid by means of, what is the bond connecting there? Which bond is connecting two amino acids? You have learned in +1. It's a peptide bond. Okay, so understand, an amino acid is joined to next amino acid by means of peptide bond. Then understand, the peptide bond formation requires energy. Okay, so for the translation, when the tRNAs bring amino acids, this amino acid should be joined. Is it not? So for that, energy is required. How that is done? Okay, that is the first phase you are going to learn. First phase of translation. It's not actually translation process. It's a preliminary step for translation. Okay, we can call that step as amino acylation. Okay, the preliminary step of translation is amino acylation of tRNA. Amino acylation of tRNA. The same step we can also call as charging of tRNA. Charging of tRNA. Okay, so all of you listen carefully. What happens in the amino acylation of tRNA or charging of tRNA? Here, at first, an amino acid combines with amino acid plus ATP. Okay, ATP is the energy rich molecule, you know. So, amino acid plus ATP forms amino acyl AMP, adenosine monophosphate, plus the 2 pyrophosphate will be released. 2 phosphates will be released. So, in the first step of charging of tRNA, amino acid combines with ATP, then this forms amino acyl AMP. It can be called amino acyl adenylate. What is AMP? You have learned adenylic acid. In short, we can write what? Adenylate. Adenylic acid can be called as adenylate. Okay, the name of this molecule. Amino acid plus AMP is amino acyl adenylate. Otherwise you write amino acyl adenylic acid or amino acyl AMP. Okay, amino acid plus AMP, the 2 phosphates. ATP means 3 phosphates, the 2 pyrophosphates released. Okay, this is the first step. In the second step, the amino acyl AMP means amino acyl adenylate is attached to tRNA. You know where it is attached. tRNA structure you have learned in the last class. Okay, so here it is attached. CCA. What is attached? Amino acyl AMP is attached, not amino acid. Okay, along. So understand the second step is what? Amino acyl adenylate is attached to tRNA. It's called a cognation. Okay, cognate tRNA. No need to study that names. Okay, so what is the result? Then we will get an amino acyl tRNA and this will be released. AMP will be released. Okay, this is amino acyl tRNA. Now this tRNA is known as a cognate tRNA, and the better term, this one you write, it is known as a charged tRNA. Okay, apparently the tRNA is not having any energy molecule with it, but when amino acid is attached to tRNA, it was with the help of energy molecule. Now this is a charged tRNA. Is it okay? So this is the preliminary step for what? Translation. That means where the amino acids are present, in the amino acid pool of the cytoplasm, number of amino acids are there. The transfer RNA are linked to this amino acids in presence of ATP. Okay, only AMP is used and that AMP also released, but this is making charged tRNAs. So a charged tRNA means it is not only having amino acid attached to it, but it has the energy to form the peptide bond. This is what is a charged tRNA. Is it okay? So suppose 2 charged tRNAs are nearby. One charged tRNA, next charged tRNA. They are close by means their amino acids can form peptide bonds without any external energy again. So understand, suppose here there is one tRNA, next nearby tRNA, here is its amino acid, here is the next amino acid. These 2 amino acids can form peptide bond without any other external source of energy. This is the importance of charging process. Charged tRNAs. I think this is clear how the charged tRNA or charging of tRNA occurs. Then we can move to the process of translation. The second phase we can discuss the process of translation. Okay, it is also known as, in simple you have learned in lower classes, this is protein synthesis process. Synthesis of protein. Okay, it takes place in the ribosome. This you have learned in very lower classes. So, an mRNA molecule. This is an mRNA molecule. It is just now transcribed from the DNA and it is processed and transported out. Now this mRNA molecule is in the cytoplasm. Okay, and in the cytoplasm there are small subunits, okay, large subunits of ribosomes. Large subunit is there, small subunit is there in the cytoplasm. Okay, and what happens is, understand, the first step is initiation. Okay, it requires number of initiation factors, IF1, IF2, IF3 like named. Anyway, for the translation, the first step is, everywhere you are studying the same terms, initiation, elongation, termination. Here also same, initiation is the first step. In initiation process, the mRNA sequence will be attached to the smaller subunit of ribosome. Okay, the mRNA will be attached to smaller subunit of ribosome. How is this possible? How can this mRNA go and clip to the smaller subunit? There are so many things to learn that all you will study in higher classes. Anyway, just understand, in the smaller subunit, there is ribosomal RNA. You have learned rRNA. Okay, here also rRNA is present. The mRNA cannot bind to this ribosome. It is actually binding to RNA. RNA will be complementary. RNA and RNA. Okay, just understand, in the smaller subunit, if it's in the eukaryotes, in the 18S subunit, 18S ribosomal RNA, there is a sequence known as Shine-Dalgarno sequence. Okay? In eukaryotes, there is Kozak sequence. This is in eukaryotes. And in prokaryotes, there is Shine-Dalgarno sequence. There are specific sequence. The triplets with the triplets. Anyway, this mRNA can go and with that anti-Shine-Dalgarno sequence, it can directly bind to that portion. Don't make it complicated. Just understand, the smaller subunit is with the RNA inside. Okay, different types. 18S, 16S in prokaryotes. Anyway, different types of small RNAs are inside. What the mRNA does, the mRNA come and bind to a specific region of the RNA, that is known as rRNA. In prokaryotes, it is known as Shine-Dalgarno sequence. In eukaryotes, it is known as Kozak sequence. That is specific, highly specific. That portion only it come and clip. Okay, so the binding becomes easier with these sequences. Okay, so understand, the mRNA now is attached to the smaller subunit of ribosome. Is it okay for you? The mRNA is now attached to smaller subunit of ribosome. Okay, the two sequences just keep in mind for the entrance purpose only. Shine-Dalgarno, then in prokaryotes, then in eukaryotes it is Kozak sequence. With the help of initiation factors, this process occurs. That means mRNA is attached to what? The small subunit. Is it okay? Then what happens? Immediately with the help of one of the initiation factors, IF3 or whatever, IF1, okay, whatever, that is not a matter for you. Here, the larger subunit comes and binds. Okay? The larger subunit of ribosome come and binds. Okay, earlier it is not as a single unit, it has separated in the cytoplasm. It is just attached only for the protein synthesis. Okay, so the smaller subunit, mRNA is attached, larger subunit come and attach to the smaller subunit. Then this larger subunit, we can divide it functionally into three regions. Okay, there is an A site, P site and E site, exit site. Okay, E site is not mentioned here and no need to mark it, just imagine 3D. The three-dimensionally imagine, A site is there, P site is there and there is one more site, E site, exit site. Anyway, this site is known as A site of larger subunit. It is amino acid site. Every amino acids are first brought to here except the first one. All amino acids are first brought to this site. So it is known as amino acid site or A site. This site is known as P site or peptidide site. At this site only the peptide bond formation takes place. So we call it as P site is peptidide site, where only the peptide bond between the two amino acids formed. Okay, so A site and P site only now our concern. There is one more site, E site. Through that the tRNAs exit. Anyway, A site, P site of larger subunit. Okay, then so this is also the machinery is ready. So mRNA is there. You know this mRNAs have its triplets. Okay. Suppose if is here AUG. AUG is here. Here is UUU. Suppose. And there will be number of other triplets forward and backward region. This is a 5' end. This is a 3' end. Okay. Then now the machinery is ready. Then who has to come to start the process? I mean, initiation means this assembly. Not only this assembly, the next step also is initiation. So what is it? In the initiation, the assembly or line-up is okay. Then what is the next for initiation? Which is the initiator tRNA. I have written here initiator codon. So initiator tRNA will come and bind to P site. Understand, always the first tRNA will be coming to P site, never to A site. Second and third onwards, everyone first come to here, then move to here. We will discuss it. Okay. The initiator tRNA. Who is the initiator tRNA? tRNA with methionine. Okay. So we are just drawing initiation here. During initiation, the assembly, first making ready the assembly, I mean mRNA, larger subunit, smaller subunit, larger subunit, everything is made ready. The next process is the tRNA with tRNA with methionine will be coming and attaching to this. Okay, with its tRNA anticodon, it is bound to this codon. Okay, so the first tRNA means initiated tRNA with methionine comes and binds to the codon in the triplet codon in the P site, not in the A site. So now the P site is occupied with the initiated tRNA and its amino acid. Now which site is empty is A site. The second tRNA, okay, will come and attach here. Okay, the second tRNA here will be A. Okay, it will bring which amino acid? We just studied. Okay. There. So understand, to the A site. Okay. That we will discuss. Initiation means this only. So initiation means what? The bringing of the first amino acid only. Okay. The second step. Our second step is elongation. I think all of you are clear with the first step. Okay. First, mRNA will be attached to smaller subunit with the help of the Shine-Dalgarno sequences. Then it can properly attach to the smaller subunit. Then the larger subunit is got attached to there. It's both the sites are empty initially. To the P site, initiated tRNA with methionine comes and attaches. That is initiation. Okay. And in the second process, elongation process. The second tRNA with its corresponding amino acid come and attach to the A site. Now both the spaces tRNAs are there with its amino acids. What is special about this tRNAs? These tRNAs are already charged. Okay. These two charged tRNAs, if they are nearby, their amino acids can form a bond. Okay. But actually this will not occur in this way, in this place. I will just discuss it. Okay. These two charged tRNAs come nearby me. It can make what? Peptide bonds. Keep in mind. So understand what happens, why the process is elongation. Then what happens is the ribosome is moving from codon to codon. After this, when both the sites are occupied, the next step, elongation, is the ribosome slightly move one step ahead. Okay? So that the next triplet comes to the A site. P site, this will be pushed. Okay. Anyway, I am just giving the second set diagram. See what happened to the next. What happened to next. That's what I am drawing now. Just keep in mind, this is the present condition. A site, P site is occupied, A site is occupied. And A site should be always empty for receiving next. So what happened? This ribosome slightly move one step ahead so that the new triplet comes to its A site. So I am drawing that thing, the movement of ribosome to the next. Codon to codon it moves. From codon to codon it moves. So what happens? Now this happened to be in the P site. Okay. See ribosome have just shifted. That means now this is in the P site. Okay. A site is empty. Okay. Here what is our next triplet is there. G, suppose. Okay. So our next triplet is there empty in the empty A site. So what's the benefit? The next tRNA can come and attack. And what happens here? See, this tRNA will be released. Okay? The first tRNA is released. Whereas this two in the P site, this is a P site. In the P site, these two I have already formed a bond, peptide bond. This is connected to previous amino acid. First and second amino acids are connected and its tRNA can be released out. So we can see the free tRNAs here. Okay, it can go and get the new one. Bring the new methionine. In between any methionine there it can bring. Okay, so understand? After making peptide bond between the first and second tRNAs, first and second amino acids, the tRNAs will be, first tRNA will be released and only the second tRNA is here with its amino acid and that is bonded with peptide bond to previous amino acid. Now A site is empty also. Then what happened? The next tRNA, okay, with its own amino acid comes. Okay, that means now this is filled. Then what happened to the ribosome next? This ribosome again slightly moved to the next position. Okay, so that it can always read new and new triplets. Okay, this is what is elongation is. So what happens during elongation? Okay, initially P site only occupied. To the A site also tRNA with amino acid come and attach. When both the sites are occupied, the ribosome slightly moves one step ahead so that its A site is with the next triplet. The previous tRNA with its amino acid remain connected to its previous amino acid so that the first tRNAs can be always released out. Used and unused tRNAs can be released out. So a chain of amino acids connected to this tRNA and empty A site. Okay, this way the elongation processes. How is elongation possible is because of the movement of ribosomes from codon to codon. Okay, then if it's clear, we will discuss the last step. What is the last step? Termination. Okay, see, for the elongation, the process also called translocation, movement from one to other, translocation. And there is an enzyme used, it is called translocase enzyme. Okay, it's another term for translocation, translocase enzyme. So, what happens? How the termination becomes possible? Tell me. You know it. Termination means. You know which are the terminator or stop codons. Suppose here happened a terminator codon UAG. If any one of the three terminator codons, ochre, opal, amber, if any one of the three terminator codons appear as the next triplet, no tRNA is specific to terminator codon. So there won't be any tRNA to come and bind and bring amino acid. So the process is stopped with the help of a release factor. Okay, release factor is usually what molecule? Anyway, with the help of a release factor, the process is stopped so that the triplet is a terminator codon, a stop codon. There is no specific tRNA for that, so the process will be stopped. That many amino acids only required for that particular protein, so that the process is stopped. Okay, so some proteins may be having many hundreds of amino acids. Some may be having few number of amino acids. Okay, so long polypeptide chain is there, short polypeptide chains are there. Okay, then this is the process of translation. When it is asked you have to describe with the help of this figure. Figure I will give it neat. Okay, with the help of the figure, with the 3 steps. The preliminary step also you want to write charging of tRNAs. Okay, and the 3 steps of translation process include initiation, elongation, termination. Once again, with a fair diagram, I will explain this. And this is the larger subunit, and this is the smaller subunit of ribosome, and this is the mRNA template, 5' and 3' end. Okay, P site, A site, two functional regions of the larger subunit. Here I have drawn in such a way that this has proceeded little ahead, in a somewhat middle way. And when there is a stop codon, it stops. Anyway, just understand here which is the tRNA present in the P site along with its amino acid. This tRNA can be released off and it can be used for the next purpose. Okay, this will be released off when this ribosome slightly move to next codon. Not in this particular position. Okay, when this ribosome moves to the next triplet, this tRNA, the first tRNA which is in the P site will be released off, so that this will come to the P site. By that time, this amino acid will be joined with this amino acid through a peptide bond, because already the charging process has done. Okay. So this is the way, and this will proceed like this. Hundreds of amino acids have to be brought to produce one protein. So it's a lengthy process. It proceeds like that. Then when it comes to a UAG, that is otherwise UAA. Otherwise, last one UGA. Any of the triplets, terminator codons. When it reaches here, there is no tRNA for that terminator codon. Then the process stops. For the initiation, elongation or translocation, then what is the termination process? Okay, and we can say chain of amino acids. This serine represents what? Its previous UCC. This alanine is for GCA. Serine is for what? UCC. Then glycine is for this GGG. Its previous triplets names are okay. So if it's difficult for you to by heart this name, you just study similar letter codons only here. Then CCC, GGG, UUU. I told to study only specific one. Okay, like this, some you by heart and that you repeat here. Okay, so when it is asked as an essay question, you have to draw this figure, and here you just to draw some triplets. Its amino acid you know. Then only you can write the amino acids in the correct sequence here. Don't write some sequences here and some amino acids here. Okay. That means this has proceeded to middle. That means these triplets have already rendered. That amino acids are here. It should be. One more thing. Just suppose here is starting with AUG. Starting codon. Here there is UAG. I am not rubbing this one. Here there is UAG. Terminator codon. But there will be some, on the mRNA, there will be some sequences before AUG and after UAG. Okay, that means the mRNA, every mRNAs are not starting with AUG and not ending with UAG or any stop codon. There will be number of sequences or number of triplets or sequences before and after. Okay, in the 5' end and at the 3' end, there are some untranslated regions, UTRs. Untranslated regions. These are required, said to be required for a very effective translation process. Before the start codon, at the 5' end, before the start codon, and at the 3' end, after the stop codon, there are some sequences which are not copied, which are not translated. Such sequences are called UTRs, untranslated regions. Okay, that only about the translations. Okay, that's all. See you in the next class.